Okay, so it is my great pleasure to welcome Mikko Tipale today as our IGM seminar speaker. So Mikko is an associate professor in the Donnelly Center and the Department of Molecular Genetics at the University of Toronto. So he received his master's degree in genetics at the University of Oulu. Not sure if I'm pronouncing that correctly, Finland, but Oulu?
Yeah.
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 And University of Oulu apparently is very close to the Arctic Circle. He then joined the PhD program in EMBL in Heidelberg, Germany. And then he did a postdoc at the Whitehead Institute at MIT, studying HSP90 chaperones in the lab of Sue Lindquist, who we unfortunately lost way too early due to cancer. Mikko started his lab at the Donnelly Center at the University of Toronto in 2014. His lab is focused on the various aspects of functional proteomics and genomics that include protein homeostasis, transcriptional regulation, and disease variant phenotyping, as well as host pathogen interactions. So Mikko received multiple awards for his research, such as Ontario Research Foundation Early Researcher Award, Margaret and Herman Sokol Postdoctoral Award, and he also holds Canada Research Chair in Functional Proteomics and Homeostasis, and was a CFAR Azrieli Global Fellow, which is one of the top awards for the early career researchers in Canada. So I also always like to mention some fun facts about the speakers. So about Mikko, fun fact about Mikko is that during his childhood, Mikko actually dreamed of becoming an astronaut. So after receiving his PhD in genetics, he applied to the European Space Agency call for the astronauts, and he was among fewer than 200 candidates selected out of initial 10,000 to become an astronaut, but unfortunately his luck ran out. But he still fulfilled his childhood dream of learning how to fly, and recently he obtained a private pilot license, which he uses frequently now. So without further ado, Mikko, take a stage. And just one more reminder, if you have a burning question, please raise your hand and ask if Mikko doesn't mind interruption, or you can post. Okay, or you can post your questions in the chat, and we'll go through them at the end of the talk. Okay, take a stage, Mikko.
Yeah, thanks very much for the kind introduction. I don't think I ran out of luck. I ran out of skill, but I think failed astronauts can still be decent scientists. I hope so. That is my hope. So anyway, okay, so you can see my screen, I hope. So I'll talk today about our work on induced proximity, about sort of an idea of forcing proteins and biomolecules to interact with each other and using that as a screening modality to identify proteins based on their function rather than their annotations. And this is kind of just one part of my lab, what we do. So like Lilia mentioned, we work in lots of different things. So not only induced proximity, but we have wide interest in the general field of protein, functional proteomics, functional genomics. But so I'll just give you a background kind of idea why we're interested in induced proximity, or kind of this kind of an approach, and it kind of stemmed from my interest and my background in protein-protein interactions and identifying protein-protein interaction networks, but it really relates to any kind of biological networks. And a lot of the biology we do now is based on the idea of guilt by association. So if you have a network of proteins or network of genes or network of any biomolecules, the idea is that if these proteins or genes associate with each other in these networks, so are more similar than average, they're much more likely to have the similar function or share the function than random proteins or genes in the genome. And that works fantastically well for many things, right? We have these networks that you can go to. You can go to different places or databases like Bioplex or for protein-protein interactions, or DepMap for genetic interactions, or you can look at other databases like GTX and so on, and see what genes are expressed in same tissues, what proteins interact with each other, and which genes have genetic interactions with each other. And then when you get these networks and look at similarities, then you can see you have lots of known proteins and genes, and there's one that is not very well annotated. You can kind of assume that this new protein or gene has, so you can come up with hypotheses as to what its function might be, simply based on correlations. And that has driven a lot of our work on protein-protein interactions, where we identify these interactors for unknown proteins and hope that these new interactors make sense. Or we identify interactors to known proteins and identify unknown proteins, and then go from there and hope that we identify the kind of have an idea of the function. But the problem is that it doesn't always work. And what we kind of the dirty little secret in the network biology field is that we just don't publish the failures, because obviously you don't want to publish your failures, but you end up picking several examples in these networks, try to follow up with them, and the one that works is the one that you publish. And the problem here is that, for example, the existing annotations are not complete, although there's an incredible amount of data out there. Still, we don't have a complete annotations for all the genes and all the proteins. And even if we had, the association doesn't give us mechanisms. So it doesn't help to know that your proteins interact with each other. It's good to know, but you don't know, is that an inhibitory interaction? Is that some sort of an enzymatic interaction? Is that just a scaffolding function, or whatever it would be? So you really need to go further than just the association. And in a similar vein, kind of the other assumption here is, we'll just check here. There's those are questions here. Yeah. So the other assumption, again, that works beautifully, is that proteins that have similar domains or the same domains are likely to regulate the same biological process. So for example, if you have a protein that has a kinase domain, it's a fair assumption that it regulates phosphorylation. Or if it's a protein that has a CRAB domain, which is a short 60 amino acid helical domain, that the protein is involved in transcriptional repression. And again, it works beautifully most of the time. And, we have these amazing databases like AlphaFold nowadays, or PFAM and SMART, where you can see what domains your protein has and have an idea already that it might be involved in, say, phosphorylation. But again, there's a lot of exceptions, and that's where a lot of the interesting biology lies. So, for example, lots of kinases do phosphorylate substrates, but there are also kinases whose main role is actually scaffolding. So there can be pseudokinases that don't actually have any enzymatic activity. Instead, they act as scaffolds, or even as E3 ligase adapters, for example. There's kinases that what looks like kinase domains, but actually catalyze a different enzymatic reaction. So, for example, ampylation, where the ATP molecule is essentially turned around so that the kinase, the domain, donates the AMP moiety instead of the phosphate. And same for CRAB domains, although they're very simple. Lots of them are involved in transcriptional repression. There's actually very potent transcriptional activators among CRAB domains, and also the ancestral CRAB domain, what seems to be the ancestral one, actually regulates meiotic recombination in vertebrates. And this is just a simple domain. And my main point here is that based on the domain annotations, you can't always predict what the function is. That the CRAB domains don't look any different based on the, for example, SMART domain annotations and so on. So you can't really say for sure that this CRAB is a repressor and this CRAB is an activator without having functional readout. So that's why we've kind of moved from this guilt by association proteomics to what we like to call guilt by function proteomics. And the idea here is that instead of identifying what proteins interact with each other, we force proteins to interact with each other and then see what happens. And in this case, we use one protein as a sensor. So this is sensor for some sort of a biological activity. We often add a tag to that sensor, and then we force different proteins in close proximity with this sensor and see what happens in that case. And this sensor can be essentially reporting on all kinds of different biological pathways. So, for example, we can have a transcriptional recorder and identify proteins that can either turn on or turn off transcription. We can have a protein recorder and look for proteins that can degrade or stabilize other proteins in a proximity-dependent manner. But really, it can be any kind of an activity of these proteins. So it could be just simple enzymatic activity, enzymatic inhibition activation, it could be protein localization, it could be phosphorylation, or really anything, any kind of a biological phenomenon or biological activity that you could have a good readout on. And often for us, the readout, we like readouts that we can use pooling for that. So we can use pool screens. So anything that you could sort with facts or you use viability as a reporter is convenient for us, but one could also imagine more complex phenotypes as well. But we think of this as when we do this at the proteome scale, this is really kind of complementary, orthogonal to CRISPR screens, for example, where you knock out genes and try to find phenotypes. So these are kind of loss of function screens, but these would be then rewiring of function screens, where we rewire proteins, bring them to new places. Why this rewiring works is that fundamental property of a lot of biology is that the specificity in biology is generally encoded by proximity rather than intrinsic specificity of these proteins. What I mean by this is that, for example, for transcription factors, you have clearly defined DNA binding domains and clearly defined activation or repression domains. And the repression or activation domain itself has no information as to what it should repress or activate. It needs the DNA binding domain to tell it to where to go, and then it will repress the target or activate the target. But if you bring it to a new place in the genome, it will actually do its job because the activation domain, for example, has no information that it needs to activate, for example, developmental regulators. And the same applies for many other phenomena pathways, for example, E3 ligases, where you have a substrate binding domain. For example, here you have a couple of different E3 ligase adapters where you have a Kelch domain or a WD40 domain that binds a substrate, and then you have the business end of the molecule that determines the specificity of these E3 ligase adapters for different E3 ligase complexes, leading to degradation of the interacting protein. But the same really works for example, chaperones and co-chaperones, where the chaperones generally are fairly low specificity, but the co-chaperones bring different substrates to them, or kinases, where the kinase domain itself, again, has some specificity, but not that much. But the targets are often determined by, the exact targets are determined by the recruitment domains, such as SH2 domains that bind phosphotyrosines, or, for example, membrane binding domains or myrcillation, for example, that brings kinases to the membrane. So really the specificity mostly is based on proximity. And the other thing why I think this is an interesting field is the emergence of the idea that we can modulate interactions with small molecules. So instead of that we can actually induce new interactions in the proteome and in cells using small molecules. And we have known these examples already for quite a while. For example, rapamycin, which is an mTOR inhibitor, it works by inducing an interaction between mTOR, so FRB domain of mTOR, and FKBP12. So it essentially induces a neo interaction. And there's many other examples of these, what's called molecular glues, that induce interactions of proteins that don't normally interact, or they can also stabilize existing interactions and just making them much more stable. So there are examples for 1433 interactions, or a lot of plant hormones also work by using inducing interactions between proteins. And then there's therapeutic molecules such as thalidomide or thalidomide analogs like lenalidomide that induce interaction between an E3 ligase cereblon and targets, therapeutically relevant targets such as casein kinase in one case. So this has led to a huge interest in induced proximity as a therapeutic modality as well. And the idea is kind of the tables have even turned a little bit, that 20 years ago, there was a lot of interest in disrupting protein-protein interactions with small molecules, but now it's almost the opposite, that there's a huge interest in inducing new interactions rather than disrupting old ones. So there's a lot of therapeutic potential also in the field of induced proximity. So So we thought that we could apply these induced proximity screens for different aspects of biology, and kind of a couple of different pathways caught our attention. The very first one was transcriptional regulation. For one simple reason was that I had a little bit of background in transcription as a grad student, so I didn't feel that it was a completely new field. But also there was at that point, there was a big question as to what do transcriptional regulators do when they are bound to chromatin, for example. We knew from annotations that there's about 1,600 transcription factors in the human genome, and we know the DNA binding specificity of most of them through large scale studies. But actually, we knew very little what these TFs actually do when they bind DNA. Do they activate? Do they repress DNA, repress expression, or do they do something totally different? So we were really interested in annotating these transcription factors, essentially using induced proximity. But we also wanted to go beyond that. So not only look under the lamppost of sort of the known TFs, but we also wanted to look at other chromatin-associated factors and just entire proteome, essentially, to find proteins that can regulate transcription either by repressing or activating transcription. And this was a project of my actually very first grad student that I recruited to the lab, Nutter, who really pioneered this effort in my lab. So the way we approached this was that we used a DCAS9 based readout. So we have DCAS9 that is fused to a plant protein called ABI1, and then we have an open reading frame fused to PIL1. And PIL1 is a plant protein that is a abscisic acid receptor, so it binds abscisic acid, and this induces an interaction between PIL1 and ABI1, which is a plant phosphatase. And then this way we can bring our open reading frames, or once they encode for proteins, so these proteins encoded by open reading frames in close proximity to DCAS9, and then we can bring DCAS9 anywhere in the genome to see how they regulate transcription. So we set up to do two different kinds of screens. One was for transcriptional activation, where we recruit proteins to silent promoter, where we have a minimal CMV, so that it's not active, but upstream of it, there's TET-O sites. So we recruit DCAS9 to the TET-O sites and then look for things that turn on GFP. And then for the repression, we had TET-O sites downstream of GFP reporter and look for things that would shut down GFP expression several kilobases downstream of the transcriptional start site. And we had stable cell lines expressing with these reporters, and they also expressed the ABI Cas9 and the TET-O guide RNA that brought Cas9 to the TET-O array. And then this enabled us to do Orpheum-wide screens, so lentiviral pool screens to identify proteins that would either turn on or turn off transcription. And Orpheum is a collection of human generally full-length cDNAs, open reading frames, and there's about 15,000, maybe 18,000 clones right now in the Orpheum we use. And then we fuse that Orpheum library to PIL1 and generated lentiviral pools for screening. We first did a transcriptional activation screen, and I'll just talk about transcriptional activation in the first half of my presentation. So we did pool screen and used flow cytometry to identify proteins that are ORFs that can turn on transcription simply by sequencing high GFP pool. And what we get out of that is that we got about 250 transcriptional activators. And what I find interesting in this kind of a system is that we have a very simple system where we have a minimal promoter, and then we have then we recruit these ORFs, but we got all kinds of different transcriptional activators. So not only did we get like sequence-specific TFs that activate transcription, but we also got components of the mediator, which is a multi-protein just a second, I'll get something to drink. Okay, a little bit better. Sorry about that. Okay, so not only did we get these sequence-specific TFs, but we also got mediator.
Mikko, sorry for interruption.
Multi-protein complex that.
I
Can you
think
hear me?
someone wants to ask a question.
Yeah.
Go ahead. If
Hi,
you
so
can.
sorry, I just want to make sure I understand the repression and the activation. So, so for the repression, why not, I guess, use I don't know, an enhancer or regulatory, like, I guess in other words, my question is why use the TET-O for the recruitment as opposed to, let's say, an enhancer region or.
So this is that's a very good question. That was our original plan, and that's exactly why we used DCAS9. Our plan was to recruit the Orpheum to promoters and enhancers and identify things that can turn on or turn off things. But turns out that it's really, really difficult to turn on gene expression with a single effector, single binding site.
I see. Got it.
Another actually, we spent like 2 years trying to activate membrane proteins, for example. And it works nicely, but it only works if you have like a BP64 or a VPR, like very super potent activators, instead of just single human protein.
I see. Got it. So sorry to interrupt. Yeah.
Yeah, no. That's a very good question. And that's actually, and I think it's relevant also, like how transcription factors work, that often single TF is not enough to activate. So you need multiple binding sites. So this sort of approximates natural case where you need multiple TFs, although in this case, we just bring one, but very likely multiple copies. So
got it. Thank you.
Yeah. Anyway, so what we get out of, so we got the mediator complex, but also histone acetyltransferase complexes with transcriptional kinases. We got polycom proteins, which was very interesting because it generally was thought that polycom is mostly a repressive complex, but we found very, very potent activators, transcriptional elongation, and so on. So it just tells us that even with a very simple system, we can find activators that activate through totally different pathways that are not only things that promote initiation, but also pause release or transcriptional elongation. So really, you can activate transcription in this system by many different ways. We then, after we validated a whole bunch of these, but one of the next things we did that we wanted to identify what are the activation domains, or what are the important regions in these proteins. So we chopped up a selection of these activators into 60 amino acid fragments with a 20 amino acid window. So each fragment was 20 amino acids off from the previous one. We fused them again to pill one, and then we just screened the same library using these fragments, and then sequenced high GFP cells to identify these transcriptional activation domains. And what we identified was these activation domains. First of all, we found existing or previously annotated activation domains. So here's just 3 examples where the gray area is the known activation domain shown that was based on previous research. So we could identify the right activation domain or right tiles that activated transcription. In many cases, we defined them much better than what was done. But it also gave us confidence that we actually find real transcriptional activation domains. But then we also found very potent activation domains in previously uncharacterized proteins that is sort of why we often do these experiments, is that we want to find these unexpected activators, unexpected repressors, instead of just annotating known proteins. So this gave us even more confidence again that our hits are real hits. They're not just random proteins, but they have well-defined activation domains within them. So this was one question that I wanted to answer, is what are the important fragments or important regions in these proteins? The other one was that we wanted to understand how they activate transcription. And one of the main important points is that TFs generally don't directly activate transcription, but they interact with different kinds of cofactors to do that. And these cofactors can be histone acetyltransferases or mediator or chromatin remodeling factors. So we turned to BioID, so proximity biotinylation, where we took several of our hits, both existing like known activators and previously uncharacterized or completely unknown proteins, fused them to this abortive biotin ligase BRA, and then identified the proximity interactors of these proteins. And that turned out to work really well. We tried APMS as well, and it works to some extent. So affinity purification pull downs, but BioID really was like shined here, that it was much better at identifying these interactions. But on the right-hand side, you can see kind of a subset of the sort of the salient points of this experiment. So the colors are just spectral counts. So the darker color means more counts of these different cofactors. And when we split them into different cofactor complexes, such as CBP-P300, which is a histone acetyltransferase complex, BET bromodomain proteins like BRD4, or another histone acetyltransferase complex NUA4, BAF is a chromatin remodeling mediator, and so on. What you start seeing is this striking specificity of activators for different cofactors. And we have these activators that interact with P300. We have activators that interact with BRD proteins, a NUA4 complex, BAF complex, and so on. So there really seems to be quite a lot of specificity, at least in this subset of activators, that they don't just talk to all the cofactors, but they have quite a bit of specificity. But if you look at, we also did BioID with the most potent activators. So VP64, which is 4 copies of VP16, this viral, very potent transactivator, and VPR, which is the synthetic activator from George Church's lab that has 2 viral activators and one human activator fused to each other, kind of a monster activator. What you can see that these activators actually interact with multiple different components. So not only P300, but also mediator and TF2D. So suggesting that that they're very potent because they're able to recruit lots of different cofactors to the promoters, explaining probably why they can activate, for example, endogenous genes so potently. So then we had these activation domains that we got out of this TAD-seq, so this tiling screen, and then we had connections to the cofactors through BioID. And at that point, we had already submitted our paper, but what happened then was that when it was under review, AlphaFold came out, and AlphaFold, like this is, now everybody knows about AlphaFold, of course, but when it came out, I think it was a beautiful example of sort of science by community, how quickly things can happen. Because when AlphaFold sort of released their code, this was, I think was late July, so this was 2 years ago. Pretty much the day after, people started tweeting that you can actually use AlphaFold to predict protein-protein interactions, not only protein folding. And this happened very quickly, within days of sort of once the code was there. And this was literally the same day that I saw this, I started testing it with our co-activators and activators. So what we did was that we just started predicting these interactions. So for example, we had these 2 unknown proteins, poorly characterized proteins, PD4 and FAM90A1, that had these activation domains, and we knew that they interact with BRD4. So we ran AlphaFold predicting how they might interact with these cofactors, and we get a very nice structure, like very nice looking structure, where the dark blue here is the fragment that we identified as a transactivator, and it's exactly this fragment that interacts with BRD4 ET, what's called the ET domain in BRD4. And when we compare this to a known structure of a viral protein that interacts with this ET domain, it's strikingly similar to that, giving us a lot of confidence that actually these interactions are real. And we can predict actually many other interactions also with other activation domains, with other cofactors such as P300. And we validated this BRD4 connection through mutagenesis, that indeed this is very likely to be the mode of binding, and it's also required for transactivation. So really showing how we can go from these associations of activation and cofactor binding to actual structural understanding very quickly, and how actually amazing it is to have like a, you can say whatever you want about Twitter, and there's many things to say about it, but when it comes to science, I think it's amazing resource how you can share science very quickly, and also of course bioRxiv, that how transformative it can be for science. So I'll kind of stop here for the transcriptional regulation. I'm happy to take questions now for the transcription part. Or we can move to, oh, there's one question.
If I may follow up again about sequence specificity.
Yeah.
So I'm wondering how might you think, the different domains, in response to what sequence they bind, then they change their effector I guess, function.
Sorry,
I mean, in other words, like because what you look at with the bioID, right, and the proximity ligation and also the AlphaFold is again protein-protein interaction. So I'm wondering how, initially, you said that yes, we know TFs, right, like they bind to specific sequences on the DNA, right, and so how might, I guess, that motif change those interactions? I mean, if you can speculate on that, because with AlphaFold you can't really, I guess, integrate, right, like
no, information. Yeah, you can't really. Yeah, I mean, my guess would be that they change very little, with a couple of caveats. So the reason why I think it's very little, because the DNA binding domain and the transactivation domain are usually very nicely split. I mean, there's examples where the DBD can actually activate or repress transcription, but generally they're very nicely split. And also the transactivation domains or repression domains are actually fairly disordered. They look like spaghetti actually, they don't quite look like spaghetti. Often transactivation domains look like a single alpha helix in the middle of spaghetti, so like unstructured, and the transactivation domain is just one alpha helix, or a couple of alpha helices. So I don't think it changes, but that said, I think there's also subtleties in terms of there's many transcription factors binding single promoters or enhancers. They bring different cofactors that will affect the local concentration of these cofactors. And we know that like transcription factors can also dimerize through DNA. So there's a lot of kind of these things that can happen. And obviously there's whole another question of phase separation condensate. But my first guess would be that it doesn't change too much, but I would be happy to be proven wrong there.
Thank you.
Okay. So, then let's move on to the next one. So this is just another example and about how we can use this induced proximity. And now we'll talk about targeted protein degradation and stabilization. So this was the second approach that we took. Actually, we started it almost at the same time with transcription, but it just took a little longer to mature. But the idea was that we wanted to identify proteins that can either degrade or stabilize other proteins in a proximity-dependent manner. So essentially using a phenotypic screen to identify proteins that would act like E3 ligases or deubiquitinases, regardless of what their annotation is or domain composition is. And the reason why we did that, honestly, the real reason was that it sounded like a fun project, but the rationale we gave to the funders was that we wanted to find regulators of new regulators of protein degradation for therapeutic purposes. And why this is relevant is that there's a huge interest in pharmaceutical companies and in biotech and generally in biomedical research in targeted protein degradation, because it's a novel modality for therapeutics and where you can degrade the target instead of inhibiting it. So the idea is that instead of, for example, inhibiting a kinase, you can degrade it and wipe it out completely, which gives you a lot of advantages. For example, these molecules being able to work substoichiometrically, and also it gives the surprising level of specificity beyond just binding to the target. So, and good examples are here what I mentioned previously, these thalidomide analogs and thalidomide that work by recruiting E3 ligase called cereblon, CRBN, to the targets, and VHL, which is another E3 ligase for which there exists potent ligands, and you can hook them up to binders of other proteins with a little linker. So these are called proteolysis targeting chimeras or PROTACs, where you have one moiety that binds a target and the other moiety that binds an E3 ligase, and those are connected with a little linker. So we wanted to find these proximity-dependent degraders in an unbiased fashion to generate a larger toolbox of proteins that could be hijacked for targeted protein degradation or stabilization, because there are over 600 E3 ligases in the genome, and only 2 of them are right now harnessed in the clinic. So this time our sensor was GFP fusion, simple GFP fusion to ABI1. So this is the part of the abscisic acids receptor system. And then we had an IRES followed by tag BFP. And then we had the open reading frames either fused to PIL1 again, like for the transcription, or we generated another library where we fused them to GFP nanobody. And then what happens here is that we can induce the interaction of these proteins, the Orpheum with the target, either through the abscisic acid system. So we add abscisic acid, PIL1 and ABI1 dimerase, and then we can see what happens to GFP. Or we directly recruit them to GFP with the GFP nanobody and see what happens to the protein. And then we use, again, flow cytometry to identify proteins that either degrade or stabilize GFP without affecting tag BFP, which is the control for expression. So we did, again, pooled Orpheum screen. So we had the stable cell line expressing this reporter, and then we infected the cells with the PIL1 or the VHH GFP Orpheum. In the PIL1 case, we treated the cells with abscisic acid and then just screened for stabilization or degradation. So these are two separate screens, although here it may look like it's one and the same screen. Okay, so what we get out of this is that here I'm plotting just the degradation screens, two different degradation screens where we have log 2-fold change. So from the bottom 10% GFP to unsorted, so things that should degrade GFP, and the y-axis is p-value. So the higher it is, the more significant the change is. And the left is the GFP nanobody screen, the right is the PIL1 screen. And what we get is a couple of hundred hits. And I highlight here known regulators of protein degradation, so E3 ligases mainly, and also autophagy regulators. And we get a very nice enrichment for E3 ligases in the screen. And indeed, for example, in the GFP nanobody screen, the very top degraders were E3 ligases. Again, suggesting that, we actually get biologically relevant hits out of induced proximity screens, not only just, garbage or some artifacts of induced proximity. We got transcriptional regulators out of transcription, and we got E3 ligases out of degradation. So this was exciting, of course, that it works, but also, we were.
Mikko, sorry, can I ask a question?
Yeah.
So on your plot, I also see like just adapters, right, to ligases, like KLH, L3.
Yeah.
It's not like UB2, which is not really E3 ligase. So can adapters also be degrade without the ligase itself, or how?
So I use like E3 ligase as a general term, right? So I meant, like, when I say E3 ligase, I also mean these E3 ligase adapters, like F-box proteins and KLH, like these BTB domain proteins. And I'll get to UB2B actually very soon. So but very good question. But essentially, they're not technically E3 ligases; they're E3 ligase adapters, but they're known to target proteins for degradation. So
okay, thanks.
But there were there are kind of two interesting things that we got out. One was that, we got unknown proteins, or like poorly characterized proteins, or things that were not connected to protein degradation. And then we got also some E3 ligases, but not that many. So we got get 20 or 30 E3 ligases in each screen, but we certainly didn't get 600, or, I think we had like something like 300 E3 ligase in the Orpheum. So we didn't get even, 100. So it was actually a fairly small fraction of them. So to test this more specifically, then we assayed them individually, all the E3 ligase we had in our library. So we cloned E3 ligase individually into this GFP nanobody construct and tested them in the same cell line just by transfection, and looked whether they could degrade GFP compared to the tag BFP control. And what we have here is 300 human E3 ligases, or these E3 ligase adapters, and their ability to target this reporter for degradation. Blue means degradation, red means stabilization, and white means that there's no difference compared to just a non-specific control, which is vanilla luciferase fused to the GFP nanobody. So there's a couple of take-home messages here. There's obviously way too much data here to kind of go through the entire thing, but there's a couple of take-home messages. First is that there's massive differences in how good they are at degrading targets when they are recruited artificially to them. They're E3 ligases that are extremely potent, and they essentially wipe out GFP completely. And then there's those that have no effect, and then there's those that actually seem to stabilize the target. So there's a lot of variation. And it also goes, it applies to all these different classes of E3 ligases. So the top three, BTB, F-box, and SOX domains, these are the E3 ligase adapters that work through the Collin ring E3 ligase system. there's a lot of variation, but so there is also variation with ring domains, for example, that then directly ubiquitinate the targets, or without the scaffold, Collins scaffold, so they also work. So this suggests that these are, there's a lot of variation here. So even in this kind of a very simple system, there's a lot of differences in how good these E3 ligases are degrading the target, which is quite surprising. And also what it shows that this is something you can't get out of domain annotations, because they're all just annotated as E3 ligases. So similar to something being annotated as a transcription factor doesn't mean that it activates or represses transcription. It can be something, it can be actually inert or not do something totally different. The other thing is that what we got out of this screen was that not only E3 ligases, but actually one of our top hits was UB2B, which is not an E3 ligase, but it's an E2 conjugating enzyme. So it's the second part of the E1, E2, E3 cascade. So it actually conjugates ubiquitin covalently to itself before E3 ligase brings the E2 to the target, leading to target ubiquitination. And UB2B was extremely potent at degrading. So here's individually we tested it, compared UB2B to cereblon and VHL, so the workhorses of targeted protein degradation. And UB2B actually worked significantly better than cereblon or VHL in this system. We tested whether how UB2B works in this case. So it has a catalytic cysteine here that is required for ubiquitin conjugation. So ubiquitin is covalently conjugated to this cysteine through a transthiolation reaction. And when we mutate this site, then UB2B doesn't work at all. So it requires the transthiolation. But also we know that E2s interact with E3s. So and UB2B has two different interfaces for interacting with them. So we mutated the ring domain interface that interacts with ring domain E3 ligases. And also on the backside, there's what's called a RAD18 binding interface. So we mutated that one, and we did it either one or the other or both interfaces. And the take-home message here is that even if we mutate both sides with 2 mutations each, we still have activity. UB2B is still active at degrading GFP, suggesting that it doesn't require E3 in targeted protein degradation, which is kind of surprising, because it was thought that UB2B or E2s always need an E3 for function, but it's actually consistent with some other data from in vitro studies suggesting that UB2B can directly transfer ubiquitin to the target in the absence of E3. We also tested another 30 E2 conjugating enzymes in the same assay, and we see UB2A and UB2B was still the best one. UB2A, which is related to UB2B, is also a very good degrader. And there's a handful of other E2s that are good at degrading, but actually most of them have no effect or have a very modest effect, suggesting again that even we know within fairly compact and well conserved E2 family, there's huge differences in their activity in this kind of an assay. Again, I keep saying this, but again, highlighting how the functions can be so different, although the domains are very, very similar. The folds are very conserved, and many of them are very compact proteins that have essentially just the E2 domain. Okay, so what about protein stabilization? And there's a question about increased GFP intensity, and I will actually get to that right now. So what about protein stabilization? So we ran the screen, of course, the same screen, and then we just screen for high GFP. And here's again the two screens for stabilization. And then if I highlight here proteins that are known to be involved in protein stabilization, we get again, we can make sense of a lot of them. So for example, we get D-ubiquitinases as we would expect. We get HSP70, for example, here. It was one of the top hits, and it's I think it's reasonable to kind of suggest that they would actually recruiting HSP70 could promote folding and also inhibit unfolding of these proteins. We have 14-3-3 proteins that are known to work as chaperones. We have a calmodulin and other kind of known chaperones for other proteins. So not only D-ubiquitinases, but also chaperones. But a lot of these actually had really nothing to do with protein stabilization itself. So, for example, there are top hits that are kinases or metabolic enzymes and so on and so forth. So what we think is happening is that there's like twofold effect here. One is that we can get stabilization through kind of just biophysics or kind of thermodynamics. So if we have a very stable protein fused to GFP nanobody, we bring it to GFP, it'll just stabilize that, just like you would stabilize a protein by fusing it to GST or some other, you know, when you express things in, or sumo or something, when you express them in vitro. So this is sort of just having a very stable protein recruited to slightly less stable protein, you get stabilization that is not related to sort of that protein's function in protein stabilization. But then we also get these stabilizers like deubiquitinases that not only work by stabilizing, you know, just by binding, but they also have catalytic activity that can promote stabilization by shaving off ubiquitin from the targets. And I showed like the next one, next I'll show you some evidence why we think that's the case. We tested deubiquitinases. Now again, we actually got a fairly low number of deubiquitinases in this system in the screen. We tested 50 of the about 100 dubs that are annotated in the human proteome. So no effector here is the no effector. In this case, we used a very unstable variant. So this is a it's a disease variant, GNMT mutant, that is barely visible when we fused it to GFP because we wanted to make the system more sensitive. So we have very unstable GFP. We fuse this to a GFP, and then we look at stabilization and use DS-red as a normalization here because it's right after the P2A cleavage site. And then what we see is that when we just fuse things to renylluciferase, we already get when we fuse the GFP nanobody to renylluciferase, we already get a 5-fold stabilization, suggesting that this really is the kind of biophysical act of binding and having a stable protein bound to the target. But then we have USP13 and several other dubs that are very good at stabilizing, that goes beyond the just regular stabilization. And we test that with specific mutations. So for example, USP38, that was one of the best stabilizers of this reporter. When we try the wild type, it stabilizes very well. But if we make a catalytic mutant, it doesn't stabilize anymore, suggesting that catalytic activity is required for the activity of USP38. For OTIB1, which is another deubiquitinase, it actually could stabilize the target in the absence of catalytic activity because we have this single mutant where the catalytic cysteine is mutated. But if we make a triple mutant that not only loses the catalytic activity, but also loses another activity of OTIB1, which is to stoichiometrically inhibit E2 enzymes, then it doesn't work anymore, suggesting that this actually, the OTIB1 requires 2 of its functions for stabilizing, so that it actually can stabilize in the absence of catalytic activity. But if we lose the second activity, then it doesn't work anymore. And what's nice when we were working on this is that at the same time, Dan Nomura's lab had identified a small molecule covalent binder to OTIB1, and they showed that you can actually target OTIB1 to other proteins using these, what they call deubiquitinase targeting chimeras or dub tags, and they can stabilize proteins using these probes. And we came upon OTIB1 through their chemical screens, and we identified it through these genome-wide, proteome-wide screens, giving us confidence that actually the results we get are relevant. And actually, Dan Nomura's lab also identified an E2-based degrader, and that was also one of the hits in our E2 screen. So again, suggesting that these are not only sort of in a genetic system, but also these can be hijacked for targeted protein stabilization or degradation. But again, for stabilization, we were interested in unexpected stabilizers, not only those that are known. And one thing we identified were these proteins called KLHL40 and KLHL41 that were very good at stabilizing the target. Why we thought KLHL40 is interesting is that those who work in the E3 ligase field, or but essentially know that these proteins should be E3 ligases. KLHL40 is a canonical BTB back-Kelch repeat protein, so it looks just like this. There's about 40 of these proteins actually that are in the proteome, and most of them are E3 ligases. But this one was a really, really good stabilizer. So for example, here you can see the GNMT reporter again. When we transfect it into cells, we can't see anything. If we co-transfect KLHL40 without any tags, nothing happens. So it doesn't just generally inhibit, say, it doesn't just generally inhibit the proteasome. if we fuse this to GFP nanobody, KLJ40 to GFP nanobody, we get very strong stabilization compared to vanilla luciferase. And then if we fuse, if we start mutating different domains, if we mutate the BTB or back domains, it doesn't work anymore. If we delete the Kelch domain, it kind of works. What do we start seeing actually? What seems to be aggregate. So what we see is that most of the protein ends up in the pellet instead of in the soluble fraction, whereas the full-length protein increases the soluble fraction of GFP, of GNMT GFP. So suggesting that the KLJ40 is actually a stabilizer of proteins rather than degrader. And this is very nicely consistent with the older studies from Eric Olsen's lab. They had identified KLJ40 in patients, or they studied these mutations in patients who have Nemelin myopathy. So these mutations in KLJ40 causes this rare genetic disease. And what they did whole proteome proteomics experiment, so whole cell proteomics in patient tissue, and noticed that KLJ40, so the y-axis is abundance compared to average knockout versus wild type. So they could see that the KLJ40 itself is down, which is expected, but also so are its two interactors, LMOD3 and Nebulin. So these are known interactors of KLJ40, and their levels also go down. So this is consistent with KLJ40 actually being a stabilizer, because if it was an E3 ligase, it would expect the interactors to go up instead of down. So this suggests that the endogenous role of KLJ40 is protein stabilization instead of degradation, and that we can hijack that stabilization function for targeted protein stabilization through these genetic means. And we think that this would be an interesting candidate to hijack this for targeted protein stabilization in, for example, myopathies where you would like to stabilize very large proteins like dystrophins and so on that you can't actually deliver by AAVs and so on. So you could actually use targeted protein stabilization perhaps in some of these disorders. Okay, so I have some minutes left here. In the end, I will kind of highlight a couple of different two more things out of the screen. One is that we had only used GFP type proteins until now, and only kind of one system. So we wanted to figure out, are these dependent on GFP, and are these dependent on this exact construct we used? To answer the second part, we generated another 9 different cell lines with GFP type proteins and tested how these effectors would work. Would they degrade all of them, or would there be a lot of specificity differences? So if we look at here, the original reported GFP ABI1 and relative GFP intensity, so these are now transfected individual, and then we just do flow cytometry to look at the effects. We can see most of these are degraders, most of them, so they work very nicely. There's a couple of stabilizers like KLJ40 and O2-1, the deubiquitinase, so we get very nice stabilization. But when we start adding different targets, then we start seeing all kinds of patterns. And the take-home message here is that we kind of three different classes, or a couple of different classes of effectors. So for example, we have here effectors that seem to be very specific to the original reporter. So for example, VHL, again, what's used for PROTAC, seems to be very finicky. It doesn't really work with lots of these targets. So they seem to be fairly specific to this reporter. There are E3 ligases that are very good at degrading targets. We call these angry E3 ligases. They degrade pretty much anything you try, so they're very good at degrading. And we think these E3 ligases would be ideal for targeted protein degradation because they don't seem to be very finicky, and they seem to have fewer limitations or fewer kind of liabilities, or might have fewer difficulties in targeting, using these for targeted protein degradation. And then we have stabilizers such as KLJ40 that seems to stabilize proteins in different compartments and different fusions to GFP. So, but this really shows that the effectors have huge differences in how they degrade different targets, but also the targets themselves are all different. So even protein fusions that are in the same compartment, such as cytoplasm and nucleus, they may have big differences in how they are degraded by different effectors. So there's a lot of compatibility issues and compatibilities between degraders and targets, likely, for example, due to availability of lysines or or the topology of these interactions and so on. And finally, we wanted to test whether these effectors can work in the absence of any tags. So we on the target. So we tested these effectors against BCR-ABL, endogenous BCR-ABL and K562 cells. We used the monobody that binds ABL, the SH2 domain of BCR-ABL, and tested whether these effectors could degrade BCR-ABL and also lead to cell death. So we generated lentiviruses that express these effectors in a DOX-inducible manner. And here you can see examples. For example, here's an effector FBXL12 that when we induce DOX expression, when you use expression with DOX, we totally wipe out BCR-ABL. FBXL15 is another very potent degrader. Whereas VHL here, cereblon there, are not very good at degrading the target, suggesting that, again, these are very, very potent degraders of proteins when you in an induced proximity system. And this also is reflected in their ability to inhibit cell growth. So here's K562 growth in the absence of doxycycline. They all seem to grow pretty well. But when we add doxycycline, all of this will have an effect because the monobody itself inhibits BCR-ABL function to some extent, but the cells are still growing. But cereblon, for example, is no better than vanilla luciferase fused to the monobody, whereas FBXL12, for example, completely wipes out cells, and FBXL15 much more significantly inhibits their growth, suggesting that we can go from this large-scale unbiased screens to identify very potent degraders of potent degraders and stabilizers of proteins. So essentially, the conclusions here are that we can use these induced proximity screens to identify effectors for different biological pathways in an unbiased manner. And our interest really is kind of to take this much beyond just targeted protein degradation or transcription, but we're really interested in using this, and we're currently using this for other aspects of biology. So identifying regulators of RNA stability and splicing, or regulators of protein trafficking and signal transduction and so on, and really interested in thinking about how we can rewire cellular signaling and cellular proteins for not only, for therapeutic purposes in the long run, but also just to understand biology and annotating proteins based on their function rather than their annotations of, domains and their interaction patterns and so on. And we hope that this kind of induced proximity screens would reveal good candidates for the development of new heterobifunctional molecules like PROTACs or molecular glues. So small molecules that would induce new interactions, or even new biologics that would do that. And also synthetic biology. So we're very interested in developing, for example, superactivators that are very good at activating transcription or repressing, or also for protein degradation and so on. And finally, these are the folks who did all the work. So Julien is a postdoc in my lab, who's our now research associate, who did most of the work that I showed. Hannah is a postdoc who joined a little bit later in the middle of the project, and she finished for example, the BCR-ABL experiments and some of the experiments in the end. And Akash was a master's student who started the entire project as it was his master's project several years ago, when he got the first sort of proof of principle concept results, and showed that we can use proteome-wide screens for identifying these degraders. And I think that was the last slide. So I thank you for your attention, and if there's any other questions, I'm happy to take them.
Thank you, Mikko. It was a fantastic talk. Very nice.
Thank you.
So did we address the question in the chat? There was a question.
There was a question about E2s.
Oh, yeah.
Yeah. So I think the E2s was
Yeah.
because of just the stabilization, because they're fairly stable proteins to begin with.
Okay. Any other questions from the audience? Go ahead.
Very, very nice. So impressive. Thank you again.
Thank you.
Yeah. My lab does a lot of like CRISPR RNAi screen, but now I'm tempted to do a proteomics screen. This is really beautiful.
I'm always happy to collaborate. So
very inspiring. So I have two questions, I guess very naive about the second part of the talk. So I may have missed it, but does the subcellular localization of the proteins that are targeted, does it matter as to how the effector responds to that? Do you see any sort of like, preference?
We didn't answer that directly. We had different targets that are different localization, but we can't and there was clearly differences, but we can't tell whether the differences are because the targets are in different localization or because the target protein is different. So we would like to, actually, I think we should do an experiment where we just put an NLS to the target and put a memory target in the and so on. So they would be exactly the same, but different compartments.
That would be nice. Yeah. And I guess follow up to this. This is my naive question. So we're using this DTAG system, right? Like where you knock in with this FKBP, right? Like tagging. So I'm just curious. So how this seems to be more attractive to use with the nanobodies. But would that be as efficient compared to, let's say, knocking in, tagging the protein?
Yes, so I think DTAG has a huge advantage of it being just small molecule based, right? So you can.
That's right.
So I think there's still a huge advantage of using DTAG.
The reversibility.
Reversibility and all that.
Right.
And we've been thinking about trying to turn these into like chemically induced degraders as well by, thinking with, for example, with the AID system. But we haven't gotten to that, but it definitely would be interesting because it seems like, based on the literature that DTAG is nice, but it doesn't always work. And then
I can tell you that we've had some targets that work beautifully and that's why I was curious about this nuclear versus cytoplasmic, right? So in that regard, this I guess because you showed with the BCL, was that the BCL? Sorry, I forget the one example you showed with the nanobody. That was BCL, right? So BCR-ABL. So I'm curious because you can also, in principle, it can also be reversible because it's DOX, right? It's DOX inducible, and so
Yeah, it is, but the time scale is sort of
the time scale. Exactly, right? That's right.
You don't
that's
get
right.
degradation in
the
a
rapidity.
couple of hours. But I mean, we see degradation like 8 hours already pretty well, but still, it does require transcription. So
that's right. Yeah. Thank you again. Very inspiring.
Thank you.
Any other questions from the audience? I guess I'll ask the last question. Actually, I was blown away by the whole my understanding of the ubiquitin ligases, because I always thought I considered like adapter proteins for ligases as kind of a non-functional, like something that needs to be there, and that ligase and E2 and adapter is just connector between E2 and the E3s, right? And I was actually blown away that some of your adapters are actually like fully functioning proteins that can degrade the targets. Like how do you think that in cells they can just function as entities by itself without like E3?
No, I think we think they actually, and we have evidence that they actually need the Collin ring scaffold. So for example, if we inhibit Collin ring scaffolds with netylation inhibition, for example, netylation inhibitor, they don't work anymore. So so it suggests that they just hijack or use the endogenous system, like cereblon and VHL are both Collin ring adapters. So
I see, but you are overexpressing them. You would think, right, that maybe if even natural E3s are there, wouldn't be enough.
I mean, they just happen to work really, really well. We don't know why, some, for example, there's huge differences between F boxes. So it may be, there may be lots of different reasons why they wouldn't work, but it's, but certainly there's a huge difference.
Can you also speculate more like how we can use it as a therapeutic, like more going into therapies, like how would we, connect like the whole induced proximity in the, in the context of therapeutics?
I think, I think that conceptually it's very, very exciting. By far the biggest problem is how do you develop compounds? And there's kind of two different ways of doing it, right? There's the heterobifunctionals, where you have a binder for one, binder for the other, and then you find a linker and bring them together, which is easier but there's a lot of issues, for example, compound size and oral bioavailability and so on. And then it's the molecular glues that are really the holy grail, that can you find single molecule that induces interactions between proteins.
And
in
it
a
has to be
non-biased
very
manner.
specific too, right? Because if it induces interactions that we don't know about, it can actually have like a lot of side effects.
Yeah.
Or it not, right?
Yeah, certainly. Yeah. I mean, so far the glues seem to be very specific because they require, you know, perfect geometry and so on. And they're, you know, they seem to be very specific, but that is the billion-dollar question in the field. And it's a literally $100 billion question in the field, that if somebody can come up with a way to identify molecular glues systematically, efficiently, that would be transformative for the field. So you know, we're solving the easy problems of biology and identifying the potential.
Well, I wouldn't say so. What you're solving is easy.
Well, that's just a lot of screening, right? And you know, then we identify effectors, but identifying the molecules that bring, you know, if we want to bring FBXL12 to another protein with a molecular glue, that's orders of magnitude more difficult. So.
Okay. If there is no more questions, thank you, Mikko, so much for the fascinating talk. Okay. Well, next time we'll see you in San Diego, maybe.
Hopefully, yes. All right.
Okay.
Thanks all.
Thank you.
Thanks for the questions. Bye.